Hello, everybody. Welcome to Miss McGuire video lectures. And today we are covering muscle tissue. This is gonna be the first video about muscle tissue, and we will cover only anatomy in this lecture. So if you are in my anatomy classes, that's all you need to watch. If you are in my anatomy and physiology class, then you need to watch part 1 and part 2. Part 2 will be about physiology of muscle tissue. Okay, so let's go ahead and begin. So overview of muscle tissue. We have three types of muscle tissue: skeletal muscles, smooth muscles, and cardiac. All muscle tissue include such properties as excitability, elasticity, extensibility, and contractility. So what it means that your muscles can be excited, so they can be stimulated. Either they are stimulated by your nervous system, or they have some special cells like your cardiac muscles. They have pacemaker cells. So those are modified muscle tissue cells that will generate electrical impulse and will excite the rest of your muscles. Or like your skeletal muscles, they need to be excited by your nervous system. And then they can contract. And contraction means there will be some movement of your muscle tissue, right? They will generate tension, I would say, yes. So contraction not necessarily means they will get shorter, right, as a whole muscle. But all muscle will generate tension, and this tension can produce movement, right? And elasticity and extensibility means they are able, as a whole muscle, as an organ, extend and without breaking those cells apart, right? Okay, so again, three major types of muscles: skeletal, smooth, and cardiac. And if you look at these histology slides, on the top, you see skeletal muscles, then in the middle, smooth, and on the bottom we have cardiac muscles, right? So let's see. Okay, so let's discuss very quickly anatomical characteristics of these three types of muscles. Now, skeletal muscles, they are very long cells, and actually each cell is as long as your whole muscle, right? So for example, if you have your bicep brachii muscle, if your whole muscle, is that long, so each cell will be, as long as your whole muscle, not including tendons, of course, because tendons that attach your muscle to a bone are made of connective tissue. It's a dense regular connective tissue, it's not muscle tissue. Anyways, the point I'm trying to make, those muscle cells are really long, they cylindrical in shape, and because cells are that long, we call them muscle fibers. So muscle fiber is the same as muscle cell, right? So if you look at this picture over here, I don't know if my video on your way or not, hopefully not. Oh, I can draw it just over here for you guys. So the skeletal muscles. Okay, for some reason my pen is not working. I wonder, okay. So the skeletal muscles, we would draw them like a long cylindrical cells, and they are multinucleated. So they have several nuclei, and nuclei kind of pushed to the periphery of the cell, right? So they are multinucleated, and they also have this lines kind of going across a cell. It's called striation. Well, striation are actually different proteins that form sarcomere, so contractive units of those cells. So we will talk about sarcomere in a second. But right now, just remember that skeletal muscles are striated, multinucleated. We call them muscle fibers because of their appearance, and they are voluntary. Voluntary means you can control contraction of your skeletal muscles, right? Now, if you move to smooth muscles, so that's this picture on the top, I'm sorry. And of course, you see like this white spaces in between. This will be some connective tissue in between your muscle cells. And also when we prepare slides, sometimes we separate the cells, and there are some spaces between shown on the slide that might not even exist in a real life, right? But we do have connective tissue between muscle cells. The second slide are smooth muscles. So you see these dark parts over here, those are actually nuclei. So don't have impression that your smooth muscle cells are kind of far away from each other and there is some extracellular matrix in between. That's not the case. You only can see nucleus of this cell, but the cell itself, right, will be kind of like this. It's a spindle-shaped cells. So here. So all this pink stuff that you see on the slide, those are actually proteins inside the cells. And we do have some connective tissue in between. Connective tissue pretty much found everywhere. So smooth muscle cells, they are spindle-shaped like this. They are uninucleated, one nucleus per cell, and they are not striated. So you see there is no this striation like in your skeletal muscles, and they involuntary. That means you cannot control contraction of your smooth muscles. And the third group of type of muscle cells are cardiac cells. So again, if you look at this histology slide, you see like a purple part. This is nucleus, right? So you have several nuclei here, right, shown. But cells are actually this kind of like pink stuff that's surrounding the nucleus. So let me draw it over here. So I would draw cardiac muscles kind of like this. They are branching cells. They have nice, big, round oval nucleus. And so here I would draw my second cell, for example, right? And you can see they have short branches. And yeah, you kind of, maybe you don't see the branches that clear, but obviously these cells do not look like a smooth muscle or like skeletal muscle, right? So they are uninucleated, one nucleus per cell. They are striated, so we do have striations. So again, those are contractile and other supporting proteins. And they involuntary as well. So that means you cannot control their action. Right? Okay, so that's our three types of muscles. So let's look at structure of individual muscle as a whole organ, and then we will get from the whole organ to the cells that make this organ. What you see on this diagram is muscle fiber. It says over here muscle fiber, but remember muscle fiber is the same as muscle cell, right? So this is skeletal muscle cell or skeletal muscle fiber. Now individual cell, it has connective tissue layer covering it that is called endomysium. So this part over here, right? This is shows sarcolemma, by the way. I'm sorry. So sarcolemma is the cell membrane, right? So you do have cell membrane. Cells cannot exist without cell membrane, right? But what would cover this muscle cell, right? I would have like this covering on top of it, right? And this covering now is connective tissue, and it's called endomysium. So I guess endomysium is just not shown here on this diagram, right? So endomysium contains extracellular fluid and nutrients. So you have a cell, and of course you cannot even draw a cell without drawing, I don't know, just want to represent it's a tube-like structure. You cannot draw a cell without cell membrane. So that's a sarcolemma or cell membrane. And then you will have connective tissue surrounding it, right? And this connective tissue is endomysium. Now, individual cells form bundle of cells. And this bundle of cells is called muscle fascicle, right? So we have individual cells and they inside the fascicle. So I guess this, let me move myself. Yeah, so they show you, so this is kind of like individual cell, right? You know it's an individual cell because look, that's a nucleus over here. And then inside the individual cells, you have lots of these myofibrils, lots of proteins fiber, but that's the whole cell. And you can see this blue nuclei because it's polynucleated. Now those individual cells are covered by endomysium. Now when you have a bundle of these cells, right? So this is the fascicle. So the whole structure is a fascicle, and fascicle is covered by perimysium. So it's around fascicles. It's allow movement of specific muscles. It's very common. You have lots of fascicles in the limbs. And then those fascicles, right? So here, like you see, that's a fascicle, is another fascicle, and another fascicle. They're surrounded by epimysium. And epimysium kind of wraps your entire muscle, separates it from other muscles, allow contraction while maintaining stability. So your muscle are pretty much bundles inside bundles inside bundles. So bundles of muscle cells surrounding by connective tissue layers. So individual cells by endomysium, then these individual cells form fascicles surrounding by perimysium, and then fascicle form an entire muscle, and entire muscle is surrounded by epimysium. Now, if you want to define what is a muscle, muscle is an organ. Muscle is the organ. So we have many muscles. So we have several organs, many organs in our muscular system. And muscle are a group of muscle cells with the same origin, same insertion, and same action. That's what an individual muscle is. Now, what is aponeurosis? Aponeurosis is connective tissue. Another name is fascia. And aponeurosis, it says here it's a tissue between skin and bones, but okay, so your muscles are connected to your bones, for example, right? They can be connected to your bones by tendons, or they be connected to aponeurosis, like we have some abdominal aponeurosis, right? For example, where we have several muscles of the abdomen connected. So muscles, the point of insertion for muscles, right? So when muscles are insert to the structure that they will move, muscles insert by tendons or by aponeurosis. So both tendons and aponeurosis are connective tissue that connects your muscles to the bones, right? I would say that. Okay, so now kind of let's look inside an individual muscle cell. So if you look inside an individual muscle cell, First, what we want to remember over here that different parts of muscle cell might have specific name. For example, instead of plasma membrane, we call it sarcolemma, because it is a plasma membrane, but it has such specific structure that it's not like your regular plasma membrane of your epithelial cells or your, for example, even nervous cells, neurons. It has different morphology, right? It has different structure and different function. That's why, we want this specific name, sarcolemma. Now, instead of cytoplasm inside the skeletal muscle fiber, we have sarcoplasm. Well, sarca means muscle. Instead of endoplasmic reticulum, we have sarcoplasmic reticulum. It's a specialized ER, right? It's a smooth ER, and the major function, one of the major functions of sarcoplasmic reticulum is to store calcium ions, right? So you can see that muscle fibers have similar organelles, right? But those organelles are not exactly the same as in your regular animal cells that we use as example of human cell. That's why they have specific names: sarcolemma, sarcoplasm, sarcoplasmic reticulum. They do have mitochondria. So you see mitochondria shown here, right? So here's one mitochondria and here's another one. Those are not different from any other cells, so they have the same name. And muscle fibers have a large number of mitochondria because they require lots of energy, high energy supply, right? Now, we already said that muscle fibers are multinucleated, so we can see several nuclei. And remember, we said they are striated. So here you see the striations. But what are those striations are? So if you look inside the sarcolemma, you already saw the nuclei, mitochondria, sarcoplasm, and then we have bundles of proteins, right? So here it's called myofibrils. So here, this one bundle of those proteins, and those bundles are surrounded by sarcoplasmic reticulum. So this structure over here, sarcoplasmic reticulum. But these myofibrils are made of different proteins, and all this name like Z-disc. Here in the middle, we have M-line. That is not shown here, but that's this the part over here, right in the middle, right? I would say this whole part here. This is our M-line. M-line. So that's gonna be a protein. M-line is a protein, myomesin. Then you have Z-disc or Z-line, and it's a complex of different proteins. And then what is attached to Z-disc, thick filament and thin filament. I mean, thin filament actins, right? So actins are attached to Z-disc, and thick filament are attached to M-line. So the thick filament shown here in the purple color. So here's the thick filaments attached to M-line, and thin filament shown in green color, and they attach to the Z-disc. And then if you look over here, in some area, we have thick and thin filament overlap. Like over here, we have thick and thin filament. We have thick and thin filament, right? Makes sense? In some area, we only have only thin filament, only this green one. And based on this arrangement, we have different bands, right? So like if you only have thin filament, you have this part of muscle you call this part of muscle fiber I band. I always remember I is really skinny letter, right? So I is skinny letter, so that's thin filament that is actin. So I band, you only have thin filament. In A band, you have overlapping thin and thick filament, right there. Now you do have some area that has only thick filament, like look over here, like look from this area to this is only thick filament over here, and it's called H zone, right? So, and how I remember it, H is almost like 2I, right? So I is thin, so that's only actin, and H is kind of like thicker, it's only myosin, because myosin is a thick filament. And A band, we have both actin and myosin, right? Oh, by the way, here you see it's M line shown over here. So whatever I show you, we have M line. So to sum up, inside the sarcomere of muscle fiber, you have organelles of a cell, regular cell, like nucleus, mitochondria. You have, instead of cytoplasm, sarcoplasm. You have sarcoplasmic reticulum. But most of the volume of muscle cell are occupied by complexes of proteins that we call myofibrils. And myofibrils are made of precisely arranged proteins. That those proteins are formed Z-disc, M-line, H zone, I band, A band, right? So and the area from one Z-disc to another Z-disc is called sarcomere. And sarcomere is a structural and functional unit of skeletal muscles, right? So between two Z-discs, right, we have actin proteins that is attached to Z-disc. In the middle, we have M-line. And what is attached to M-line are myosin protein, right? Thick filaments. Now, area where we only have thick filament is called H zone. Where we only have thin filament is called I band. And where we have overlapping thick and thin filament, we call it A band, right? And then those sarcomeres repeats, right? Because muscle cells are very long, right? So obviously you have many of these structural units in a line. And because we have over here kind of overlapping proteins, when you look under the microscope, it looks dark. So those dark lines are actually your A bands, right? That's why the skeletal muscle cells look striated, because of this actin and myosin overlapping within A band. That makes sense? I hope it does. Okay. So here you can look at structure of sarcomere in a little bit more details here, right? You can see structure of myosin or a thick filament. It's only show you a portion of the thick filament, and you know it's kind of it has this myosin tail, and then flexible hinge region and heads. So you see lots of heads coming out from this myosin molecule, right? So here are the heads, and they like in pairs. Here 2 heads, here 2 heads. Also, we have ATP binding site, because we will need ATP for muscle contraction, and we will have actin binding site, because we need myosin to be attached or bind to actin. But we will talk about it in physiology part, in part 2 of this lecture. And if you look at actin, it's kind of like two strings of protein. So the actin itself, right? So you have these green proteins in a, so it's, you know, it's kind of like two strings like this, right? So that would be actin. And then each actin has this line over here. This is another protein, and this is called tropomyosin, right? That's a tropomyosin. So here's the name. And at some parts of tropomyosin, you have another protein over here that is troponin. So actually your thin filament, it's shown over here. It's your actin and troponin tropomyosin. So we have actin shown here in green, right? So that's a green, shown here in green, actin subunits. And then we have tropomyosin, and then we have troponin right there. And see this dark green side over here, this is a binding site for myosin. So actually this actin binding site and this binding site for myosin, they need to bind together, right? So that the whole kind of purpose of this structure is, and the whole theory behind it, that if we have actin and we have myosin, and myosin heads gonna bind to actin, and they're gonna push these actin molecules towards the center of sarcomere, and sarcomere will be shorter, will become shorter, and the whole muscle will contract. But you can also see that tropomyosin blocking this binding site. So in next lecture, when I will explain you this sliding filament theory, or it's an explanation of how our muscles contract, we will see that for myosin to bind to actin, this tropomyosin need to be removed from the binding site. And for that reason, calcium need to bind to troponin. Right, so all this will make sense when you start learning the physiology of muscle contraction. And of course, on the top, you can see Z lines, and what attached to Z line is actin, M line. You see, this is Z line instead of Z disc. So you can use both Z line or Z disc. I just like line. I don't know why we need disc and line and band and zone. You know, should be maybe a little bit consistent with naming, right? So Z line, M line, actin attached to Z line, myosin attached to M line. And remember, I band, I is skinny letter, only actin. H zone, it's a, you know, kind of like two eyes almost. You need to put two eyes and connect them in the middle. So it's like thick letter, you know. And again, you guys can come up with mnemonics that works for you, right? You don't need to remember this stuff. You can just, maybe you can just remember that H zone is just a thick filament. That's it. But sometimes little, you know, kind of association helps you to remember better. And then A band is actually, it's the whole length of the myosin. And in some part, myosin will be overlapping with actin, in some part it will not, but A band, you see, it goes as long as the myosin molecule. And in some area over here, we have overlapping. And then myosin itself is pretty thick. And remember, striations that we see this A band because it's a thick filament, plus we have some overlapping parts with actin and myosin. That's pretty much right. And don't forget that sarcomere is a functional and structural unit of muscle fiber. It's a region of one Z line, from one Z line to the next Z line. We will go into this neuromuscular junction in more details when we talk about physiology. Right now, I just want to mention that skeletal muscles need activation from your nerves, from neurons, in order to contract. And this area where motor neuron and muscle fiber meets, we call it neuromuscular junction. So it makes sense. It's a space between your neuron and muscle cell, right? And you know, junction means they kind of like come together. And there is a space. It's not direct physical contact between neuron and muscle, right? And it's a synapse. Because we have synapses between two neurons, we have synapses between neuron and muscle cell. And what's gonna happen, your neurons, when they fire, right, the electrical impulse will come to the this neuromuscular junction, and several steps will happen. But the kind of like the final step at this neuromuscular junction would be the release of special chemicals that we call neurotransmitters. And specifically between your neuron and muscles, you release acetylcholine. So it's ACH. That's a neurotransmitter called acetylcholine. When acetylcholine is released, so you can see this red particles here represent acetylcholine. Acetylcholine will bind to its receptor. So it's acetylcholine receptor. And when acetylcholine binds to its receptor, it will allow sodium to move inside a muscle cell. And this will start the chain of events that will lead to muscle contraction. So it's pretty much in muscle contraction. First, we need activation of muscle cell by motor neurons, and it will be done by release of acetylcholine. Acetylcholine binds to its receptor on the muscle cell. This binding will cause sodium influx, sodium goes on, and then we have series of events that eventually allow your muscle to contract, generate tension, and move a load. So for example, move your bone. Okay, so we did not cover any of the physiology, right? Remember, this part 1 is only for anatomy. So now let's talk very briefly about cardiac muscles. They found only in heart. They also striated. They have sarcomeres. And another thing that they have, see, here we have two cells and they connected through this junction, and this is called intercalated disc. And it allows cardiac muscle cells to contact in a wave-like pattern. So ions will move from one cell to another cell, and when ions move, that's electricity moving, and it will allow muscles to contract all together. Instead of one muscle contract and relax, another muscle contract. So you want your heart to contract as a whole unit. Within this intercalated disc, you will have desmosomes that will anchor the ends of cardiac muscle fibers together. Right, so here you see intercalated discs, right? So you have this gap junction. So allow ion to move from one cell to another, but you will have also these desmosomes that will hold cells together. Cardiac muscle cells do not require activation from the central nervous system because they have orthorhythmic cells. Orthorhythmicity is the ability to some cells, not all cells, but some cells, specific cells that we call pacemaker cells, to activate the rest of muscle cells to contract, right? So pacemaker cells control contraction of the heart. So instead of nervous system, you have pacemaker cells. So pacemaker cells pretty much gonna do the same job that your nervous system does for your skeletal muscles. So for skeletal muscles to contract, you need to receive signal from your neurons. For cardiac muscles to contract, you need to receive signal from pacemaker cells. Now, smooth muscles are found in walls of hollow organs, and they spindle-shaped fibers. We already talked about it. No striation. However, they do have actin and myosin. It's just not organized in these precise sarcomeres, and we don't see this striated appearance. They have dense bodies. Those are analogous to the disc that anchors a thin filament, right? And smooth muscles, it's shown here that they activated by autonomic neurons, right? So that means they are not voluntary. So those autonomic neurons gonna release neurotransmitters and your smooth muscle cells will contract. And sometimes it doesn't require, it doesn't require your central nervous system to do it, because those autonomic neurons can be found in the local plexuses, like myenteric plexus, right? So it will be the whole network of neurons inside the walls of your stomach, for example, that pretty much will regulate contraction of your smooth muscle without central nervous system. And central nervous system can regulate it. It can speed up or slow down. Okay, and just very briefly about development. So these muscle fibers are so long that they obviously cannot divide. I cannot imagine the cell, microscopic, but that long cell to start dividing. What are you producing? Another huge cell? You wouldn't have enough resources to do it, right? So muscle cells do not divide. The number of muscle fibers are predetermined by your genetics. So how many muscle fibers I have, it's in my genes, right? I mean, there is variety. Some people just have more muscle fibers. However, muscles still can grow bigger. So if muscle cells cannot divide, how your muscles are grow bigger? And they obviously do. If you compare yourself with a little, infant or with a baby, obviously their muscles are way smaller, right? So your muscles somehow grew bigger. And if you start exercising, your muscle get even bigger, right? So what's going on? At the same time, if muscle cell cannot divide, what's going on? But actually you do have special cells. It's called satellite cells. They similar to myoblasts, and myoblasts is like cells that are capable of division, right? So those satellite cells shown over here. So this is a satellite cell. So when your muscle grows, these satellite cells incorporate itself. So you see how it's kind of became a part of your muscle fiber, and it's deposited its nucleus even inside, right? So, this, because you have this nucleus, that will facilitate protein synthesis, facilitate repair and growth. So if you see over here, this is untrained muscle. So that's my muscle, right? Even I'm trying to do more exercises to train my muscles. But, that you have, good amount of nuclei, but when you start training routine, you will see the fusion of satellite cells, right, with muscle fibers. And now look, you increase drastically the amount of nuclei, and plus there's other organelles like mitochondria, cytoplasm, right, inside this satellite cells. So you kind of like adding all the ingredients, And now you're ready to synthesize more proteins. And when you synthesize more proteins, your cell became, the cell itself became bigger. It's the same cell. It didn't divide, but it goes through the hypertrophy. So cell increases in volume, right, and size. And then if you stop your training, right, so detraining, you can see that muscles can return back to their original size, but you still have so many extra organelles to begin with. So it's going to be easier to retrain the your muscles. So now you kind of skip the step of fusion of satellite cells. You already have all the organelles, all the machinery needed. And remember, you even have in satellite cells, you have ribosomes, right? As I said, endoplasmic reticulum. Those are just, cells that have all the regular organelles of a human cell. So you pretty much can skip this fusion and go straight to hypertrophy. Now, this process of your muscles to lose their volume, right, they get smaller, called atrophy. So if you don't use your muscles, they will go through atrophy. If you train them, they go through hypertrophy. Hyper means bigger. Atrophy means you're losing this mass, right, and volume. My goodness. Okay, now, so your muscle cells can use satellite cells to heal from injuries as well. However, if injury is, a pretty drastic, if it's a, big injury to your muscle cells, then your muscles will heal by fibrosis. And this is where we replace muscle cells by scar tissue. So if for example, if you cut yourself and it's a deep cut to your muscles, you might have scar. And this scar is a repair of your muscles by scar tissue instead of by muscle tissue. Right. And if we're talking about smooth muscles, we have pericytes. Peri means kind of like around, right? Or next to, not around, right? So those are stem cells that generate smooth muscles. So we have satellite cells in skeletal muscles, and we have pericytes that generate smooth muscles. And smooth muscles are the best to healing. So they heal better because we can generate new smooth muscles. Those pericytes can divide. Cardiac cells and skeletal muscle cells, they rely mostly on fibrosis when it comes to repair damaged cells. Now, and couple diseases over here, actually just one disease we're gonna look at, muscular dystrophy. It's a group of inherited muscle destroying diseases, and muscles go through atrophy at the end. But first they kind of get enlarged, and muscles look even getting bigger, right? But they enlarge due to fat and connective tissue deposits. And fat and connective tissues, they cannot contract. So when you replace your muscle cells with connective tissue cells, you might replace the volume and the mass, but you cannot replace the function. So you lose the function of your muscles. But let's look what's going on with muscular dystrophy. So this is your muscle cell, right? So this is inside your muscle cell. And inside you have a special protein. It's called dystrophin. That's interesting, right? So that's a dystrophin. And what dystrophin does, it connects cytoskeleton, right? So it's a kind of cytoskeleton is the structure inside your cell that give your cell shape, right? And stability, allows stuff to move inside. But this dystrophin connects cytoskeleton to a protein within a cell membrane. So here's the sarcomere, that's a membrane, and there is a whole complex, right? So this is, like huge protein over here, and it's called dystrophin associated complex. So dystrophin is connected to this intermembranous protein, and this complex connects to extracellular matrix, right? Like basal lamina. So you have some proteins in extracellular matrix outside of the cell. So this is outside of the cells. And this gives a stability to muscle membrane. Now, people with muscular dystrophy, it's an inherited disorder. That means there is some mutation within their genes, and this dystrophin is not synthesized. So when they do not have dystrophin, then you don't have anything to hold the cell membrane in place. And sooner or later, this membrane start breaking apart, right? So it's it's bends, right? There is some holes inside, and then some proteins, it's called CK proteins, they will escape. Maybe some, for example, calcium will move in, and so the cell cannot keep homeostasis anymore, right? It losing the stuff, it gaining the stuff that is not supposed to gain. So finally, cell will die, right? And this will cause, if some cells die, you replace them with fat, then you're losing function, right? So there is different type of muscular dystrophy. The most severe is Duchenne muscular dystrophy, and you can see that different type of dystrophy can affect different muscles of your body, right? So you don't need to memorize many of the steps. Maybe just remember that Duchenne muscular dystrophy is the most severe. And severity of this disease is that your diaphragm is affected, because your diaphragm is a skeletal muscle. And if diaphragm cannot function, that can cause the respiratory distress and respiratory failure and death, right? So, so here's our Duchenne muscular dystrophy. Most common and severe type. It's inherited, is sex-linked. It's carried by females and experienced in males. So it's 1 in 3,500 births. And it's lack of dystrophin, as we already mentioned. So victims become clumsy and fall frequently, usually die of respiratory failure in their 20s. And there is no cure. We use viral gene therapy or infusion of stem cells with the correct dystrophin gene, and it shows promise. So what we're trying to do, because they have this gene that produce dystrophin defected, right? There is a mutation in this gene. So if we will somehow introduce a healthy gene to these patients, we hope that then, muscle cells start synthesizing dystrophin, right? So, and this is the diagrams that show you the effect of this disorder, right? So it causes skeletal muscle degeneration, and the muscles, stop functioning. So the person now cannot move his or her body, so they will end up in a wheelchair, right? And because the diaphragm is affected, it can cause the respiratory failure. It can be inflammation of cardiac muscles and cardiac and respiratory dysfunction. And unfortunately, there is no cure. So it's a very, very sad prognosis for these kids. And it has some indirect action as well. So hopefully the gene therapy will advance in the near future, and we will be able to help these kids to live healthy and long life. Okay, so that's it for this part. So again, it was only anatomy of muscle tissue. Thank you for watching, and I hope it was helpful.